All right. Welcome back, anatomy and physiology students. So last week we were covering the anatomy and physiology of the spinal cord, and this week we are continuing with our focus on the central nervous system by moving into the anatomy and physiology, structure and function of the brain and brain stem. So in this first video, I'm going to be going over some general overview related to the structure of the brain and brainstem. And then in the next video, I will go into the specifics of all of our separate parts, these named regions of our brain and brainstem. So here is a general look at all the different parts of our brain and brainstem that we'll talk about in this chapter. But like I said, in this first video, we're just going to go over some basics. And in the next video, I'll actually dive into the specifics about the medulla oblongata, pons, mesencephalon, aka midbrain, thalamus and hypothalamus, cerebrum and cerebellum, etc. So start off with, we have named different regions of our brain based on how they show up developmentally in embryos. And this also gives us a bit of an idea or a look at how these structures of the brain developed evolutionarily as well. So all of the central nervous system starts out as this continuous single tube called the neurocele, essentially what is full of cerebrospinal fluid. And that neurocele starts to differentiate into these early structures, these little round scoop-looking things that are separate from the part of the neurocele that stays linear, which is going to become the spinal cord. These kind of bulges that come about early on in development represent early versions of these regions of the brainstem and cerebrum, right, your brain. So we have the rhombencephalon, aka the hindbrain, which is also sometimes referred to as the lizard brain evolutionarily, as this represents regions of our brainstem that were some of the earliest structures in the development of a central nervous system. This middle region, the midbrain or mesencephalon, and which is sometimes referred to as the rat brain or the early mammal brain. And then this most superior or anterior region, depending on the organism, the prosencephalon, the forebrain. And this is going to turn into some of the higher order processing centers that we associate with primates and humans. Right? This is a much later evolutionary development that we only see in those specific groups of organisms, again, primates and humans. So further on throughout development, these regions are going to further separate and develop and start localizing what become regions that we recognize in a fully developed brain. So the hindbrain, this rhombencephalon, is going to split into the myelencephalon, which will develop into the medulla oblongata and the fourth ventricle, as well as the metencephalon, which will develop into the cerebellum and pons, as well as portions or regions of the fourth ventricle, since that is kind of in between all three of these structures. The mesencephalon basically stays the same as far as it is only developing into a single structure of our brainstem, our midbrain. And then the cerebral aqueduct is just the space for cerebrospinal fluid to transmit between the fourth ventricle and the third ventricle, which is going to show up in our diencephalon. So the prosencephalon, this forebrain, is going to split into the diencephalon, which is going to become your thalamus and hypothalamus, as well as the telencephalon, which is going to develop into the cerebrum, what we typically think of when you think the brain, that big, bulbous, wrinkly part, as well as the lateral ventricles, becoming the ventricular system, aka the equivalent of the vascular system for the body. The ventricular system is what is containing and transmitting all of the cerebrospinal fluid to transmit nutrients and remove waste within our brain. So these different regions are considered the brain vesicles. And like I said, they show up throughout development as this neurocele is starting to specialize into the structures that we recognize as fully developed parts of our brain, and also gives us that look at our evolutionary history. Essentially, the more inferior or posterior is representing kind of the earlier versions of a central nervous system of a brain that would have come about. And as we move more superiorly or anteriorly, we get some more recent and higher order processing versions of the brain. And so those terms are further explained here with the brain vesicles, but we also have terms referring to those folds and valleys, those peaks and valleys that we see showing up in our brain structures. So some terms to look out for are going to be the gyri, sulci, and fissures. Or the singular version would be a single gyrus, a single sulcus, and a single fissure. These come about simply due to the pressure of the space that the nervous tissue has as it is developing, ends up pushing that nervous tissue into these folded structures, which we have also found through research and observation tend to correlate with the amount of processing power that a brain has. Essentially, the more folds seems to have a direct correlation with a higher level of processing power. So again, those gyri are going to be the bumps or the peaks, and then the sulci are going to be the indentations or the valleys. And so some gyri and some sulci are going to have specific names to them. And then fissures are just like super deep sulci, which all have specific names to them. And those are typically separating specific regions of the brain, such as the lobes or hemispheres. So I mentioned earlier a little bit about the ventricular system. So we have ventricles, which is different than our vesicles. Okay? So the vesicles were these early regions that are going to develop into structures that we identify by name as part of the neural tissue. Ventricles are the vascular system of the brain. Right? This is what is housing a majority of the cerebrospinal fluid. These ventricles all become continuous with the central canal of the spinal cord. And this gives the central nervous system its own separate, protected and contained version of a vascular system. Right? And we'll talk a bit about the blood-brain barrier and the blood-CSF barrier that kind of help keep these systems separate. But that CSF is there to support and protect the brain and the spinal cord. And so in the brain, we have four major areas where we have the cerebrospinal fluid kind of flowing through to supply the brain and remove waste. So these are our lateral ventricles, which show up within the cerebrum. We have the third ventricle, which essentially is within and around the diencephalon, so within and around your thalamus and hypothalamus. And then we have the fourth ventricle, which is within your brainstem, in between the pons, cerebellum, and medulla oblongata. And this is a continuous system. They all connect with each other. We have the interventricular foramen, which is the name for these canals that connect the lateral ventricles to the third ventricle. And then we have the cerebral aqueduct, which kind of runs through the midbrain in order to connect the third ventricle with the fourth ventricle. And then that fourth ventricle can just flow straight into the central canal, right, to continue throughout the spinal cord. So that is our ventricular system. We have a cool model of this in lab, which I may or may not have shown already at this point, but keep a lookout for that or ask about it next time you see me. And so this slide is just putting all of that into words for you, somewhere to easily track what I was discussing. And beyond these ventricles, within the cerebrum and brainstem, we are going to have cerebrospinal fluid flow out into some of the spaces within our cranial meninges. And so we have the same meninges that we were discussing with the anatomy of our spinal cord. We have a pia mater directly adhered to the nervous tissue of the brain. We then have the arachnoid mater, more superficial to that, and then dura mater as the most superficial meninge. And these are just different connective tissue layers. They're going to provide some slight differences in their function as far as support and protection for the brain. But there is cerebrospinal fluid as well as capillaries and veins all throughout the spaces within these meninges. And so we get a look at these meninges here, right? So, and you can see that vascular system kind of penetrating through. But our dura mater for the brain, slightly different than how it was with the spinal cord. The dura mater is composed of two layers. We have the periosteal cranial dura, which is directly adhered to the bones of the skull. And then we have this meningeal cranial dura, which is technically separate in lots of regions around the skull. They're going to be fused together, essentially the same connective tissue structure. But in some regions, they can separate, and that creates a dural sinus. which is essentially space for cerebrospinal fluid and the veins and capillaries of the vascular system to exchange nutrients and waste. Some of those dural sinuses are going to penetrate into the structures of the brain, and those create dural folds, which have slightly different functions for each of our major dural folds. Let's see, or I guess skipping a bit ahead here. Then, like I said, deep to the dura mater, we have the arachnoid mater, named because of the spider web looking structures that show up with the connective tissue of that arachnoid mater. And there's going to be, similarly to what I was describing with the dural sinuses, lots of vascular tissue. The arachnoid mater is highly vascularized. And then that vascular tissue is essentially bathed in cerebrospinal fluid to, again, help with that exchange. And then pia mater directly adhered to the brain itself. So, or the neural tissue of the brain. So when we do our brain dissection, you can get a sense of kind of the difference in the structure for these things. You'll see, hopefully, we don't always get brains with dura mater on them, but if we do, you'll see that really tough, almost leathery dura mater as the outermost layer. We can cut that even separately without cutting into the arachnoid mater and pull that off. You'll then see that the arachnoid mater creates like a thick spider web all the way around the structure of the brain that makes it look really smooth. And the arachnoid mater does not follow the folds of the sulci and the bumps of the gyruses, of the gyri. And so it creates a smooth structure all the way around the brain. And then we can pull away that arachnoid mater, and then you're left with pia mater, which does adhere directly to the neural tissue. And so it will follow those gyri and sulci. So there's something cool to look out for when we get to do those dissections in lab. And again, here we have that put into words for you. And so a quick note on the blood-brain barrier, as I may have mentioned it already in some of the previous lectures and or in lab, the cerebrospinal fluid of the brain, that vascular system with the ventricles and the central canal, so the cerebrospinal fluid of the entire central nervous system, rather, is kept separate from the blood of your vascular system, of your cardiovascular system. And that is mainly being protected and upheld through this blood-brain barrier. And so for most of the blood vessels throughout your body, you have endothelial cells that are closely connected to each other, but will have little tiny gaps in between each endothelial cell, and that allows for the much easier release of and exchange of materials between the tissue surrounding the blood vessels and the blood itself. But the endothelial cells of the veins that are in capillaries that are in the brain or anywhere around the central nervous system have these tight junctions, which prevent a lot of that free movement or exchange, as well as these special parasite cells, not to be confused with PARA parasites, but pericyte cells. And then lastly, the end feet of the astrocytes, those glial cells, that are all creating extra layers of protection, preventing that free movement or exchange of just any random molecule traveling in the blood or any random molecule that might exist in the CSF. And so that creates this extra specialized protection. And so there is still exchange, things like oxygen and glucose and carbon dioxide that need to get to the neurons of the central nervous system to fuel them, again, glucose and oxygen, and then waste and carbon dioxide that needs to get out of the central nervous system to be able to be released from the body. Those things are still able to freely move, but the directionality of that movement is much more closely monitored and controlled. And so only certain larger molecules, such as psychotropic drugs, are able to get across that barrier, and most other large molecules are going to be completely blocked and unable to get into the CSF. So there are going to be regions of our central nervous system where the blood-brain barrier doesn't exist in quite the same structure. And so we'll talk a little bit about that in the coming up slides. We have a blood CSF barrier, which is just specialized in a different way to allow for other types of molecules to get across that barrier. Things like hormones, right? In regions where we have glands that are closely related to our endocrine system, like near the hypothalamus, the pineal gland, the pituitary gland, near the pineal gland, and near our choroid plexi. These are all regions where you wouldn't see the same level of layers of protection that we consider part of the blood-brain barrier, and instead you get this blood CSF barrier, which is just a slightly different structure to allow for more exchange of very specific large molecules. Again, mainly hormones, right? So speaking of that blood CSF barrier, one of the regions where we see that show up are the choroid plexi or choroid plexuses. And the choroid plexus is a region with these specialized ependymal cells that produce all of the cerebrospinal fluid of the central nervous system. So we have choroid plexuses show up in a couple different places. In the floors of the lateral ventricles, the roof of the third ventricle, and the roof of the fourth ventricle. So all of the cerebrospinal fluid of your central nervous system are produced in these locations. And again, that is coming from these specialized ependymal cells. And so these ependymal cells don't produce the same level of protection that the blood-brain barrier did, but instead produce this blood CSF barrier with a bit of a different level of exchange going back and forth between them. And then this diagram is just giving you a look at that overall flow of how CSF is moving through the entire central nervous system, from the choroid plexi through the lateral ventricle, the third ventricle, the fourth ventricle, into the central canal, and then even how those are exchanging into places like the subarachnoid space or the dural sinuses in order to allow for some exchange with the cardiovascular system. And so that is where I will stop with this part of chapter 14. And so in the next video, I'll start getting into some of the specifics about the various structures of our brainstem and brain. So I will see you in the next one.